I'd like to share with you an application of DILIsym where we used our mathematical model to predict low frequency or idiosyncratic drug-induced liver injury in clinical trials. There have been multiple applications of DILIsym throughout the clinical development pipeline. In fact, it was designed, the model DILIsym, to assist in evaluating hepatotoxicity risk for compounds throughout the pipeline. And the focus in this presentation is more on the clinical side, the clinical trial side, in order to predict a low incidence of drug-induced liver injury, also known as idiosyncratic DILI. And in this application, we are combining predictions of exposure, hepatic drug and metabolite exposure, with mechanisms of hepatotoxicity to make these predictions of injury. And to do so, we are employing our representation of inter-individual variability, also known as SimPops. Let me first provide a brief overview of the DILIsym modeling simulation platform. We include 4 species in DILIsym: the human, the rat, the mouse, and the dog, and population variability is included, or inter-patient, inter-animal variability is included in each of these species. We also have really essentially all of the primary cellular processes included in drug-induced liver injury. And this diagram is sort of a summary of the key submodels and how they interact. And starting from the upper left, you can see we have our submodel of drug metabolism and distribution that then predicts the exposure of the parent and any metabolites within the liver. And that interacts with the various pathways, mechanistic pathways of hepatotoxicity, including oxidative stress, mitochondrial dysfunction, lipotoxicity, reactive metabolites, and intracellular bile acids. These mechanistic pathways have some interaction as well. Ultimately, they're all capable and interact with the hepatocyte life cycle to induce either apoptosis or necrosis. There's also a feedback with the innate immune system, which can exacerbate in some situations or ameliorate in other situations the injury. The innate immune system also drives the regenerative response post-injury. There's also a submodel of biomarkers of injury that captures the dynamics, as particularly is comparable with what is measured in the clinic. That is the qualitative description of these submodels. In order to establish the quantitative connections, we've used a number of exemplar compounds that have been established to have mechanistic interactions and drug-induced liver injury in a variety of different ways. And that allows us to, like I say, establish the quantitative interactions within a submodel and between submodels. DILIsym is a compartment-based model that employs ordinary differential equations. It also includes a form-to-function connection, wherein we continue to track mass balance despite potentially the loss of hepatocytes in predicted injury situations. So back to our application, a PPAR gamma agonist, which was the first in its class of the thiazolidine diones for treating type 2 diabetes. It ameliorates peripheral insulin resistance and hepatic insulin resistance and reducing glycemia. It was available on the market for a brief period of time until hepatotoxicity was reported in 2% of the patients. Hepatotoxicity had not been detected in the preclinical studies. The magnitude of the injury was substantial enough that the medication was withdrawn from the market due to this idiosyncratic hepatotoxicity. And subsequent studies have identified multiple possible mechanisms that could have contributed to this hepatotoxicity. The focus of these simulations, however, is exclusively on bile acid-mediated toxicity. A little bit to expand on how bile acids may elicit hepatotoxicity, particularly the troglitazone effects. You can see listed here, this is kind of the hepatocyte within its native environment. You can see blood flow from left to right, and NTCP is the transporter that's responsible for bringing bile acids into the cell. BSEP is the transporter that's responsible for canalicular transport out of the cell, while MRP3 and 4 are responsible for basolateral export of bile acids from the cell. So bile acids enter the cell via NTCP, like I say, and can be exported via either the BSEP or the MRP transporters. Troglitazone, upon entering the cell, gets metabolized such that proportionally there is significantly more troglitazone sulfate, the metabolite, than the parent compound troglitazone. Nevertheless, both have inhibitory properties against both BSEP and MRP3 and 4, which cause bile acids to accumulate and leads to the potential for hepatotoxicity. Troglitazone also inhibits NTCP, somewhat mitigating the accumulation of bile acids within the cell. So in order to consider the inter-individual variability, we employed our SimPops, otherwise known as simulated populations, wherein we vary key parameters, and in this case, you can see, given the context here of the PBPK submodel, the bile acid homeostasis submodel, bioenergetics, hepatocyte life cycle, and biomarker submodels, the key variables that we combined within a known range of values were included in the PBPK, so the exposure side, as well as the bile acid mechanistic side. And so we made a large number of simulated patients, in excess of 300, and various parameter values here, combined all those in various ways, and we were able to predict a range of responses based on a range of biochemical properties combined in unique ways, establishing a unique individual, accounting, like I said in the beginning, for that inter-individual variability. So the first set of simulation results are given here in this slide, where you can see that the simulated patients, N of 331, were dosed either 200, 400, or 600 milligrams of troglitazone a day. And for the 200 milligrams, there was very little increase in ALT, but dosed proportionately, there was an increase, so that there was a higher frequency with 400 milligrams and an even higher frequency of ALT observations increases in 600 milligram dose simulations. And so we're establishing here that there is interpatient variability in the response, and that there's a relatively low frequency of response, some of the simulated patients having pretty substantial ALT maximum. So taking these simulation results and translating them from graphical form into tabular form, you can see here are the columns representing the frequency of hepatotoxicity for each of the simulated dosing groups, 200, 400, and 600 milligrams, compared with what was reported in the publicly available data for the clinical trials. And you can see that the predicted incidence of where in the clinical trials the patients ranged from 200 to 600 milligrams based on tolerability. In combining the frequencies at each of the various ALT thresholds, the predicted frequency is really nicely consistent with the observed frequency or the reported frequency from the clinical trial data. While bilirubin, and Hy's law cases were not necessarily reported, the incidence of jaundice is somewhat consistent with the predicted levels of bilirubin, as well as the incidence of Hy's law. So we were able to then take those simulation results, which clearly showed a nice comparison in the low frequency drug or idiosyncratic drug-induced liver injury, and relate that back to those particular parameters that were varied amongst that simulated population. And in doing so, we were able to determine some factors that really seem to indicate susceptibility to troglitazone hepatotoxicity. And these are listed here. So those in bold indicate that they could potentially be susceptibility factors. That includes the troglitazone sulfate biliary clearance. And that was the primary metabolite that was included in the simulations. Bile acid biliary excretion, FXR mediated feedback regulation, body weight, and LCA synthesis in the intestinal lumen. So really these include these potential susceptibility factors, kind of an intersection between exposure to the drug, the underlying genetics, as well as the exposure. And by exposure, this includes the microbiome, this includes dietary effects. And so there's a really interesting potential here for drilling down, and I might add, the potential to identify patients who, prior to dosing, who could have been susceptible, and determine those that are appropriate for dosing. So to summarize, DILIsym was utilized to predict this low frequency or idiosyncratic DILI that was observed and reported for troglitazone. This work has been published and is available in the literature. We also identified a number of possible susceptibility factors that might have been used to separate responders from non-responders prior to dosing. And this predicted low incidence of troglitazone hepatotoxicity was predicted exclusively by the bile acid transport inhibition mechanism, and which is really quite interesting. I should also point out that this publication was supported by a comment from members of the FDA Office of Clinical Pharmacology in this same journal, that in pulling out a specific quote from this commentary, I'll read it aloud. "We look forward to future efforts to apply this model for prediction of hepatotoxicity that has not been clinically observed." So with that, I'd like to conclude, and thank you for your time.